All right, so now for the main feature today. So our previous provost, Michael Gottfredson, made a decision during the recession that what we were going to do as a university is take advantage of the talent pool across the country that many universities weren't able to take advantage. So he focused all the resources he could muster to hiring the best young people in the country. And several of those you've heard from, and today we're going to hear from one we're very proud of, Aaron Esser-Kahn. So Aaron received his PhD in chemistry from UC Berkeley. His bachelor degree is from Caltech. His research interests include microvascular materials, carbon sequestration, and synthetic immunology, which we're going to hear about today. Once again, just as a measure of how successful this program of hiring the best young people has been, Aaron has already received the 2014 DARPA Young Faculty Award. DARPA is Defense Advanced Projects Research Agency. Close, anyway. He won the 2003, or he earned the 2003 NIH New Innovator Award, and the 2013 Hellman Faculty Fellowship. And he continues something that we're very proud of here at UCI by combining research fields to create a new field. And so today he's going to tell us about his work to combine chemistry and immunology. So welcome to Aaron.
Am I on? Okay. Let's see if my iPad stayed connected. Okay. Thank you, Ken, for the introduction and for the opportunity to speak to all of you today. I'm going to talk about about one half of what my group works on, and as Ken alluded to, we have sort of two different areas of interest, both focused on really large problems and what chemistry can bring to bear in pushing those problems forward, thinking about new ways of approaching them. And so today I'm going to talk about our work in synthetic immunology, where what we're trying to do is sort of the key bits, these last two words in my title here, so vaccine and design. What we'd like to be able to do is use chemical tools to design an immune response. And so I'm going to talk about how we're thinking about that problem, why it's important, and what specifically we're doing to address different challenges. So the reason that I'm interested in this and the reason that my group is interested, I think it pretty much be summarized on this one slide. So this is a slide from the CDC, and on your left here you see all of the morbidity rates, or the number of people that were infected and died from a specific disease before the introduction of the vaccine, and then the number after the introduction of the vaccine. And the numbers vary quite a bit depending on the disease, but probably two that I'll highlight here are pertussis, where you see about 200,000 people, and now only 20,000. And then the one that I think is most interesting to me is actually varicella. And so varicella is chickenpox. So who here in the audience, when they grew up, got chickenpox? Yeah, and you can see that my group, because they're the youngest people here, very few of them raised their hand. And that's because varicella, the chickenpox vaccine, was introduced in 1995. And overnight, it went from, on the geological time scale, I should say, but within 5 years, it went from 4 million people getting chickenpox every year to basically 500,000 people. And in the next couple of years, people expect that number to drop even more to basically 500 or so per year. So between 1990 and 2014, varicella was essentially wiped off the face of the earth. And the reason for that is because people developed a way to tell the immune system how to find varicella and track it down. And there's still a great need for more basic discovery tools in this area, right? So chickenpox, while annoying and somewhat infectious, all you really get is some scabs. But of course, these are much more serious diseases. So tuberculosis, where in fact about 1/3 of the planet is a carrier of tuberculosis, and 1 million people die each year, down to HIV. where there's 30 million people infected and about 1.6 million deaths. And none of these diseases currently have vaccines because we don't know how to tell the immune system how to track them down. And of course, the bit that got the most press here was the disease that currently is wreaking havoc on the United States or the world is Ebola, right? So everyone's scared because Ebola is a new disease. It's highly infectious. It has a particularly nasty outcome. And so we need ways to develop new tools to talk to the immune system so we can tell it, hey, here's Ebola, please get rid of it. So the reason that it's been such a challenge to develop these types of vaccine responses is that literally nobody knew how to talk to the immune system or how the conventional vaccines that we developed actually performed that task. So no one knew at the basic mechanics how it worked. You can imagine the way that you understand how a car works. So there's an engine, there's wheels, there's force that's transferred throughout the material. No one understood vaccines at that level. They understood the input, which is to say the way that you create the vaccine, and then the output, which is the result, the fact that there's no more disease, but they never understood the mechanics, and that's what we're really after. And as that began to occur, so in 1997 there was a critical discovery made by a guy named Ruslan Medizov where he found the first protein receptor responsible for the way that a vaccine works. And what you can see is that on this graph here in red, these are the number of vaccines that have been developed in the course of human history on this axis, so 70 total. And you'll see that right around 1997 there's an inflection point in the growth curve of vaccinations. And that's because now understanding the molecular mechanism of how a vaccine works or how an immune response works allows you to get better and better at developing that type of response. And the chemical, the word I'm going to use to describe these chemicals that talk to the immune system is adjuvants. It's literally Greek for helper. So going from empirical knowledge to then fundamental understanding created an inflection point and a growth curve that allowed vaccine development to really take off. And just to contrast this, here in black, these are the number of small molecule drugs that have been developed in the course of human history. And here's the axis, so it's still 2 orders of magnitude more, but what you can see is that a similar growth curve, there's actually an inflection point around 2000 where the rate of growth of vaccines now exceeds the rate of growth of small molecules, though of course there's many, many more small molecules being developed. So this was particularly interesting to me as we thought about this area. So, as I'm sure many of you here have heard, there's a lot of myths going around about vaccines, so I wanted to take one slide to sort of address all of these, so I'll call these the tricks of vaccines. But these are just 3 sort of central myths that seem to be propagating through the community. One is that vaccines are very profitable and that people like to give them because they make a lot of money. That is just not true. Vaccines are the least profitable part of the pharmaceutical industry. In fact, they make less than 1% of the total profit in the industry, and most of the major pharmaceutical companies have shut down their vaccine development divisions because they don't make enough money. So no one's making money on vaccines, and the reason is because they're very, very cheap. They're pennies per administration, and then of course, usually 1 or 2 administrations cures the disease for the rest of a person's life, right? And so there's not much money to be made in actually making somebody well. There's a lot of money in making somebody slightly better than they were. And so vaccines are not profitable. The other thing that's come about a lot is that vaccines are not safe, that they cause autism. And I would say that vaccines are safe, and that safe is always a relative term. When you're considering that you're getting 1 case of side effects out of 1 million people, if you consider other things that you do on a routine basis, like take Tylenol or drive a car, both of those are much, much more dangerous than getting inoculated with the vaccine. So vaccines are actually one of the safest things that human beings do on a large scale. The last thing is herd immunity, which is sort of the piece that makes people think, I don't need to get vaccinated. I don't need to get my kid vaccinated because, of course, we don't need 100% of people to get protection from a disease, right? And that really depends on the disease, to be honest. So there are certain diseases where, say, 95% protection will work. But there are other diseases like pertussis in particular, which I'll show you some data on, where it doesn't work. So herd immunity is a myth. It really is disease by disease. It depends on the transmission rate and the way that the disease incubates. And to be honest, nobody knows that statistic until people don't vaccinate their children. So the one that's most relevant to California is whooping cough. So in fact, California had the largest outbreak of whooping cough in 2010 in the last 50 or 60 years. And what you can see here in this chart on your left is that there's a direct correlation between the percentage of children vaccinated and the number of incidents of whooping cough. And these outliers here, of course, this is not perfect data and it's statistical, but the point is that the outliers here usually occur from caches of people with much, much lower vaccination rates. So you may say there's 97%, but that 3% may all live in the same town and go to the same school, and so those are the ones that end up getting whooping cough. And so you can see that on the second graph on the right here. This basically shows the percentage vaccination rate versus the cases per 10,000 children. So the lower the rate in the given county, so if it's in vogue, say, for people within your school to not vaccinate their child against whooping cough, then the lower the percentage of vaccination, the less people who are protected, the higher the number of cases of pertussis. So it tracks directly with the percentage in this case. And that's because pertussis and whooping cough is one of those diseases that tends to hang around and then infect people when they sort of least suspect it. And then, of course, because it's passed and transmitted in the air, it's very, very infectious, so it can spread very rapidly. So those are the three tricks, if you will, of vaccines, just to sort of get those points hammered out. Vaccines are incredibly safe. They're one of the safest things you do. They're not, no one makes money on them, unfortunately. And third, you have to get vaccinated. Everybody has to get vaccinated in order to protect themselves against the disease. There's no such thing as herd immunity. So with that somewhat dour note, I'll switch to talking about why we're interested in the problem and what we think we can bring to bear with new solutions. So I'm going to talk first about the basics of vaccinology and innate immunity, how we're thinking about talking to the immune system. Then I'm going to tell you, sort of highlight two projects that my group is working on. One is in developing new types of adjuvants, which we call coupled immune agonists, which is to say that we're taking the way that these molecules present themselves to the immune system and we're rearranging them using chemical tools. And then the last part I'll talk about is another area we're interested in, where we're doing what chemists call caging a compound, which is to say we're creating a mask and then revealing it later on so we can program the response that we want. We can essentially push a button and turn on the immune system remotely with a laser beam. And so that's sort of the two areas that we're working on, and I'll try and motivate why we think both of these are important. So again, the challenge is getting these types of responses. If you can do it for a disease, you effectively wipe it off the face of the earth. But the challenge is, of course, figuring out how to do it, right? And then the other challenge is that you have to be able to design these responses for different age groups. So, 2-year-old's immune system is very, very different than an 80-year-old's immune system. And so you want to be able to design vaccines that work for both young populations, where you can give one shot, say, even before they leave the hospital, so they're immunized for the rest of their life against any number of different diseases. And at the same time, you want to design vaccines for aging populations, where the challenge is eliciting stronger immune responses and to different pathogens and different diseases that affect only those who are maturing in age. So that's the idea. We're trying to get those types of responses across a number of different people. And so to kind of summarize, this is what we know so far, right? This set of different responses. And you can kind of imagine a plane of all the knowledge about an immune system and how to manipulate it and work with it. And so these are sort of what we know right now. Today's vaccines mostly developed against viruses and bacteria. But at the same time, you could go out into the wilds of all of the different human beings on the planet. And what's really fascinating is you can find people whose immune systems already know how to do things like resist getting cancer, right? So there are people who are already immune to different types of cancers. They produce antibodies against these cancers. They resist forming lymphomas, and they essentially never get cancer. At the same time, there are actually people on this planet who are completely resistant to HIV, right? There are a few people in Africa, you can pull their blood, you can find the antibodies that they've developed against HIV, but you don't know how to create that response in every person. There's people resistant to heart disease, there's people who are resistant to getting type 2 diabetes, all of these in one form or another COPD, all of these in one form or another are immune diseases. They all require the immune system to operate, and talking to the immune system is the key aspect of either developing a disease-like state or developing or staying normal. And so our idea is we'd like to take what's known about vaccine design here and what's known currently in the population and connect them. So we want to create links, knowledge links between what's known about people who are resistant to different diseases and what's out there right now, or what we know right now, and sort of eventually the goal is tomorrow or in 10 or 15 years, we can have vaccinations and immunotherapies for a host of different diseases. So we can tackle traditional viruses, or we can tackle cancer, or we can tackle COPD, each of those requiring a different set of notes or communication with the immune system. So the question is, of course, how do you fight a disease, right? There are sort of two different approaches to this, and this is my metaphor for thinking about why you would want to talk to the immune system. So one approach to fighting a disease is you have your immune system on one side, the pathogen on the other. They're duking it out. So of course, if you want to win a fight, the best way to do that is to cheat, right? Which is to say, you want to give one side an unfair advantage. And for us, we'd like to give us that unfair advantage, right? I think that makes the most sense. We're not trying to make the pathogen smarter and stronger. So we want to figure out how to give the advantage to the immune system so that it can do what it does best. And so one approach is to cripple your enemy, right? So in this case, I just sort of illustrated this as removing the sword from their hand. And this is how antibiotics and drugs work. So antibiotics and drugs, what they do is they find a weakness within the pathogen, and they exploit that weakness, effectively slowing the pathogen down and crippling it so that the immune system can then come in and do what it does best, right, and eliminate the pathogen. So the challenge with that, as we've seen in recent years, is that pathogens tend to get smarter. They tend not to just take it lying down, so they may go away for a little while, but eventually they come back. They come back stronger and harder and tougher than they were before. So this is basically drug resistance, right, which can be summarized in the form of MRSA, which is multiply resistant Staphylococcus aureus. And staph basically is resistant to every antibiotic we can produce at this point. And so developing methods of enhancing the immune response is the only sort of solution we have left to treat some of the most resistant diseases. So the other option is, instead of crippling your enemy, you can make yourself smarter and faster and better, right? So one thing you can do is you can educate yourself, and that's how vaccines work, is that you basically teach the immune system how to effectively combat a particular pathogen. And so our goal, right, is to improve the immune system by making it tougher and smarter so that it's able to combat these things in an effective manner. And in order to do that, we basically have to send the immune system to school, right? And so this is the idea. We want to educate the immune system about specific pathogens. And so I'll sort of talk to that point. So how do we start a dialogue with our immune system? How do we educate the immune system effectively? And so to summarize, I'm going to go into a little bit more of the specifics here. I'll talk about different cell types, but and I'll kind of go back and forth between the metaphor and the innate immunity here. So on one side, we have adaptive immunity, which is the response that you're probably most familiar with, which is the antibodies that you eventually produce against a given pathogen, as well as the T cells, which travel around your body and essentially destroy foreign pathogens. But that adaptive response is actually controlled by a part of your immune system which is far older. It's called the innate immune system, and it's the part that usually when you were in maybe 8th grade or 10th grade biology, you heard about, but no one ever actually knew what it really did, right? So they were like, well, there's neutrophils, and there's granulocytes, and there's these things called macrophages, and they sort of they move around and they pick material up. But it turns out that in fact, what people discovered around 1997 or 2000 is that the innate immune system influences or directs the response of the adaptive immune system. So it's the gateway through which the communication occurs. And so then the question is, immunologists usually stop here, right? They're like, well, there's these two parts of these cells. There really isn't anything else to the picture. And then when I came into this field, I asked this question, or started reading. I said, what is the input to this system? If the cells are making this decision, if they're controlling these antibodies, what is actually controlling the cells? And the answer, lucky for me and lucky for chemists everywhere, is of course that it's molecules, right? Because at the end of the day, any biological system accepts molecular information as a way to make a decision. And so the chemical modifiers, the inputs to the immune system, is what we're particularly interested in. And manipulating those modifiers to direct ultimately the antibody and T cell responses that are necessary for vaccine. So this is our language. These are the molecules we use to talk to the immune system. We do so specifically through these types of innate immune cells, which are called antigen-presenting cells. And I'll mostly talk about dendritic cells today as the major class of antigen-presenting cells. So this is not just this is not something that only I do at UCI, right? I think the reason that people were asking me earlier, how did you end up here? I think the reason is that essentially I came 3 or 4 years ago. I pitched this idea to the faculty, and it really resonated with them, because I think I exist right here along with my group at this interface between chemistry and innate immunity, and you can see that there are a lot of other people at UCI that exist at this interface, right? So you can take the interface between gaseous reactions and spectroscopy, and then you get atmospheric chemistry out of that, and you get Sherry Rowland. You can take chemical reactivity in microscopy, and we just saw some examples of that in Castle and Ara Apkarian's work. Understanding how molecules work. And you can take synthetic polymers and muscle proteins, and you can get people like Professor Zibin Guan who are working at the interface between those two areas. So when I came here and I told the people at UCI chemistry that I wanted to do something at the interface, they said, that sounds great. That's exactly where we live, right? And that's just not a very conventional thing. I went to a lot of other departments and I told them, I'm a chemist, but I want to work with innate immune cells. And they said, why in God's name would you ever want to do that? And the people in the chemistry department here said, of course you would want to do that, because that's where the future is. The future is at the interface between two disciplines, not where we already know, already have a good picture of what's going on. So the idea is to go back to the concept of what we're after here. We're after sort of designing an immune response. And the metaphor I'll use for this is essentially you want to educate the immune system or talk to it in the same way that you do track down a criminal, right? So my metaphor is that the way that you educate the immune system is essentially like a wanted poster from the Wild West. So I have Billy the Kid up here as an example of that, and I'll sort of go through the key elements here. So if you want to track down a specific pathogen or a specific disease, you need to think about how you're going to identify it and then eliminate it. And so this is the same thing as sort of wanting and finding a criminal. So you have a couple of different elements on a wanted poster, right? You have the problem, which it turns out Billy the Kid's name is William Henry McCarty. But of course, if you were ever to ask anybody to look for William Henry McCarty, you would never be able to find him, because he goes by a code name. He goes by an alias, which is Billy the Kid, right? So you actually need to know his alias in order to find him and track him down and eliminate him. And the other thing you need to know is a method of identification. So a way to figure out exactly where he used to be. So last known location, right? Where to begin looking. So in this case, that was Fort Sumner. And then the last thing you need is a method of apprehension, so a method of capture. And so in this case, for a criminal, that's dead or alive, right? So if you take this idea and look at how you would use it to create a vaccine, this is how you would think about doing this for a pathogen. So I'm going to talk a lot about yellow fever virus vaccine, and that's because it's a model example of vaccines, and it turns out to be the best one ever developed. And so the disease is known as yellow fever virus. The alias, the code name in this case, and I'll get into this in detail as we go through how this works, but the code name is these different receptors, and they're called toll-like receptors. And so TLR stands for toll-like receptor. So that's the code that the immune system uses to initially flag and identify yellow fever. And then in order to track down all of the yellow fever and eliminate it from the body, it uses an antigen, which is a series of amino acids, a polypeptide, which it can develop an antibody against and then eliminate the disease from the body. And then the last thing it does is it sort of marshals its forces. So this is a little bit more complicated than just dead or alive. So the immune system has to pick, do I want to produce antibodies? How many different antibodies do I want to produce? Do I want to create a different type of immune response where I put cells out with antibodies on them that go out and effectively eradicate the disease? And so immunologists categorize these as different types of responses. So they're called Th1, Th2, Th17. There's a whole host of these different types of responses that are necessary to sort of effectively eliminate the pathogen. So the reason I picked yellow fever virus is it's a very effective wanted sign, right? In fact, so one shot of yellow fever virus vaccine, if you're a kid, basically causes you to be immunized for life. So 40 years of immunization for one shot. And so this code, this particular code that they used, TLR2, 7, 8, and 9, turns out to be one of the most effective codes known. And so you can see this now reflected in the way that immunologists measure these same types of things. So this is the magnitude and persistence of the different types of responses that happen when you inoculate somebody with a yellow fever virus vaccine. So yellow fever virus vaccine, this is now my cartoon. I moved it from from this green guy. Same basic idea, but now trying to get it closer to what it looks like at a molecular level. Still, now each of these represents different elements of that code. And so what you can see is that it's a very short and quick innate immune response. These are the blue and yellow lines here. And then a much longer T cell response. So these are CD4 T cells, CD8 T cells, and then a very long-lived IgG and IgM response, which are the antibodies that eventually persist in your body and remember the pathogen and allow it to be eliminated when it reoccurs. And so all of that information is encoded in these molecules that are existing on the outside of yellow fever, these yellow circles and in this case, orange squares. So I'll sort of walk you through how the immune system makes this decision to get to this end result, which is the production of antibodies that cause you to be immune to a particular disease, as well as cytotoxic T cells. So it uses two different pieces of information. So it uses the antigen, right, which is what people are mostly familiar with, which is the identification marker. And here I've cartooned that antigen as being this series of different colors indicating the different amino acids in the polypeptide. And through that information, in order to get to the antibody response, it communicates through these dendritic cells, these antigen-presenting cells, which are responsible for processing the chemical information and then presenting it to the T cells, which then direct the B cells and the cytotoxic T cells. And it does this through these two types of responses, right? The Th1 type response, the Th2 type response. And in each of these cases, there are a number of different cytokines, which are the proteins that direct the T cells, ultimately giving you the antibodies. And so immunologists have unfortunately come up with very difficult to remember nomenclature. They just named them in the sequence they found them. And so it doesn't really mean anything, but IL-2, IL-5, IL-6, each of these has a specific message that it sends to the T cell, communicating to it, present this antigen, and then tell the B cell to produce this certain amount of antibody, right? And the decision to produce those cytokines is made inside of the dendritic cell. And it is accepting chemical information in the form of the antigen, which is this, again, these series of different colors indicating the polypeptide, as well as the code that it's accepting, which is the agonists of the toll-like receptors, the agonists of the pattern recognition receptors. So an inactive dendritic cell goes to being active when it accepts chemical information and dimerizes a protein, which then sends a signal allowing the cell to make the decision to produce cytokines. So it's a sort of a little bit more Rube Goldbergian than you would like, but it is the way that it works, right? And so the key is really controlling the association of these receptors and controlling them in a manner that is not just turning them on and off, but effectively it is like a language where you activate them in a spatial and temporal manner. So you activate them one at a time across certain locations and at certain times. And so the inputs to this system, the chemical information that goes into it, are these small molecules, polymers, and macromolecules, right? And that's where the chemists, like me, come along. Because what we can do is both create new versions of these molecules, as well as control and manipulate these molecules in order to affect this response, and then to program the antigen-presenting cell ultimately to get the type of response that we want to develop the vaccine response that's necessary. And we can do this with different types of antigens, but the antigen is essentially along for the ride. It doesn't really encode any information in and of itself. It's just there. It's what the immune system ultimately uses to flag the pathogen. But the code, the information of self versus not self, that's all contained within these toll-like receptor agonists, these PRRs. And so again, to give you an example, this is how yellow fever virus vaccine works. So you get this balanced Th2 and Th1 type response that's so critical for eliminating the pathogen. And these different cartoon shapes, these represent different toll-like receptor agonists. And so what the yellow fever virus vaccine does is it activates TLR7 and TLR9 in a type of dendritic cell called a myeloid dendritic cell, or sorry, in a plasmacytoid dendritic cell, and then it activates TLR2 and TLR8 in a type of dendritic cell called a myeloid dendritic cell, and it's that combination of activation of all 4 of these different receptors in sequence across 2 cell types that allows you to communicate to the immune system, elicit this effective response, and eliminate this pathogen for the rest of your life, right? So the code is the orange triangle, orange square activating the DCs, and then the orange square and the yellow circle activating the myeloid dendritic cell. And so you can think about this as basically a code of molecular agonists. If you punch the right buttons in the right order, you get the right immune response that you want at the end of the day. And so that's what my group is interested in, is figuring out how to punch those buttons and in what order in order to elicit the type of response that we want. So if you can develop a code and use the code effectively, then you get the type of response that you want. And this turns out to be true of almost every empirically developed vaccine. So every empirically developed vaccine activates a palette of different toll-like receptors. And so each to a different level and to a different degree. And you can see a few of them on this slide. And it turns out to be true of many different pathogens as well. So viruses, bacteria, fungi, protozoa, all of them communicate through different toll-like receptors, as well as a series of other pattern recognition receptors. So each one has a unique code that the immune system uses, and some of those codes are more effective than others, right? And so yellow fever virus vaccine is a highly effective code. Just the virus on its own activates the immune system very strongly, allowing you to eventually create antibodies and eliminate the pathogen. But the challenge for new diseases, things like tuberculosis, malaria, HIV, or as we mentioned earlier, Ebola, is that they're very sneaky. And at the same time, they don't have a lot of molecules that allow them to encode a response, and they're very good at hiding their antigens from the immune system. And so they can basically eliminate this information from our normal immune response, and that's why we get infected with them. That's why they persist in our body, because the immune system doesn't know how to eradicate them, it doesn't understand what they are, and it doesn't know where to look for them. And so the idea is that we can take this knowledge, right, that we know about how people already respond to these different types of diseases. So if we can find people who are resistant to Ebola, we can figure out what antigens they're already producing, and then we can use those antigens as an effective way to vaccinate against the response. So the antigens are known for, say, Ebola, they're known for malaria, they're known for HIV. The challenge is in finding the effective code that elicits the type of response you want. And the reason is because there is no way to read that code. It exists transiently only when those molecules interact with the immune system. And so in order to make a type of code, to understand those codes better, what we're trying to do is synthesize them in our lab, create molecules that elicit different responses to begin to understand how to activate and probe, and develop code names that allow us to trip off the immune system and push it towards effective responses against things like Ebola. So that's what we're after. We're trying to design our own codes. So we're taking what's known about things like yellow fever. We're abstracting the principles of yellow fever in a tool that chemists usually call retrosynthesis. So the idea is you take what you already know and you work backwards from that. And so we're trying to design molecules that have the same type of response as yellow fever virus vaccine, but are synthetic, are much smaller, and we can understand every component that we put into them. So we can build our own codes using the tools of chemistry. And the way I'm going to refer to this in the next couple of minutes as I talk about a few examples of what we've been doing in this area. These are the receptors, and these are how they communicate through the dendritic cell. So we've now gone all the way from the entire immune system and zoomed into a single cell, and now within that cell there are these specific receptors that are responsible for activating the immune system. And there's a lot of information on this slide, but the key element you need to know is, again, these are the inputs, these are the molecular inputs to the system. The thing thing we're going to measure is the NF-kappa B response, and NF-kappa B response is the master transcription factor that turns on all of these cytokines and interferons that are responsible for eventually programming and getting you the type of B cell responses and T cell responses that you want. So all of it really comes down to tickling these specific receptors in an effective manner to elicit the type of NF-kappa B response that you want. So that's the introduction, that's the idea we're after, and I'll tell you two stories here, two vignettes about what we're doing to explore those different areas. So the first is in coupling together these different immune agonists. So to go back to my metaphor of the wanted sign, there are lots of pathogens that it's already known how they effectively talk to the immune system and how they work. And so we in order to begin this process, we figured if we were going to mess around with these codes and these code names, we would pick a virus that people already knew how its code worked and see if we could understand how manipulating the molecules that are part of that code allowed us to elicit a better response. So in this case, we used herpes, which happens to stimulate TLR2 and TLR9, so toll-like receptor 2 and toll-like receptor 9. And that's where we began. And so this was a paper published in 2006, and they showed that herpes activates TLR2, which happens to be on the cell surface of an antigen-presenting cell, and TLR9, which is in the endosome of the dendritic cell, and that in then turn activates and stimulates the rest of NF-kappa B and these interleukins that are necessary for the immune response. So using this idea, this principle of working backwards, we abstracted what was necessary in the herpes virus. And remember, the herpes virus is very big compared to the molecules that we're using. So a herpes virus is usually measured in megadaltons, which you could imagine is like megatons in terms of weight. And the molecule we're making is measured in daltons. So it's 10 to the 6th less in size. But we think we can take the same basic principles and abstract them, turn them into a smaller molecule, and use that molecule as our effective code for activating the immune system. So we deconstruct the pathogen in order to reconstruct its code. And so what we did is we took a known agonist of toll-like receptor 9, which is called CpG, and a known agonist of toll-like receptor 2, which is called lipotecoic acid. And all we did was chemically couple them together. We just took them from being two molecules and made them one molecule. And we defined the distance that these two were allowed to be away from each other. If the activation of receptor 2 and receptor 9 is necessary, and it's necessary that they do this in a sequence, a spatial and temporal sequence, then coupling them together will allow them to perform that function. So we asked the basic question, does proximity affect activation? And so these are just the building blocks for those of you who are the chemists in the audience. Lipotecoic acid, the business end of lipotecoic acid, the part that we're after, is this long lipid tail that activates TLR2. And in a TLR9 agonist, it's a piece of single-stranded DNA, and the portion that activates toll-like receptor 9 are these CG repeats, and the reason it's called CpG is because of these CG repeats, which bind to toll-like receptor 9 and activate it selectively. So we use these because they had chemical handles that would allow us to manipulate them and synthesize a molecule where we coupled the two agonists together. And so here's just a slide showing this data. So what we did is took a bifunctional linker. We linked together lipotecoic acid on one end and CpG on the other. We created our chimera, where now we have a piece of DNA and a piece of lipotecoic acid bound together in the same chemical structure. And so we refer to this as the LTA-CpG heterodimer, which is to say, and the underscore just indicates chemical linkage here in terms of how we think about it. We did a lot of analysis because, of course, if you're going to publish this in a journal, you have to prove to everybody that you made what you said you make. Otherwise, it's not science. So we did a lot of analysis, but the key element, what we were really after, is we wanted to activate toll-like receptor 2 and activate toll-like receptor 9 and then measure the amount of NF-kappa B response that we got. So the amount that we turned on this master regulator protein, which indicated that we were turning on the immune system. So the way that we did this is we used a very straightforward assay, and so it's called a raw blue assay. And essentially what happens is you use these cells, and the more NF-kappa B that's activated, they turn the solution from red to blue. So the more NF-kappa B activation, the more immune stimulation, the bluer the solution becomes. And so what we found when we did an NF-kappa B assay is if you look at just CpG on its own, so this is just activating toll-like receptor 9, you see very little stimulation. When you mix the two molecules together in equimolar ratios, that is to say there's the same amount of each molecule now present at these concentrations, you see a tiny bump in that activation, right? But herpes, of course, activates much, much stronger than these two molecules on their own. So what happens when we chemically couple them together? Well, the reason the rest of the slide is blank is because when we couple them together and make a single heterodimer, we completely change the way these two molecules interface and interact with the immune system. We, at much lower dosages, immediately turn on NF-kappa B and end up getting much higher NF-kappa B responses than just the molecules unattached to each other. So chemically coupling them using the tools of organic chemistry, we completely change the way that these two molecules activate the immune system, mimicking the way that herpes does it. And my postdoc did one last experiment that I thought was particularly intriguing. He ran this against the strongest known agonist of the immune system to date, lipopolysaccharide, which is so strong, in fact, that it can't be used as an adjuvant in a conventional vaccine because it causes sepsis. And so what he found is that, in fact, our molecule is stronger at higher dosages than lipopolysaccharide is. It activates NF-kappa B at much higher amounts than just LPS on its own. But the twist is that it has a very different dose-response curve, right? So at lower concentrations, it's much less active than LPS. And this is great if you want to develop a vaccine, because what you want is to turn the immune system on locally at a single point when you get the inoculation and not let the molecule diffuse everywhere and activate a broad response that leads eventually to septic shock. So changing the way the molecule worked and changing its dose-response curve enabled us to create a new molecule which is potentially useful as an adjuvant for promoting vaccine design. So Rock's in the audience. This is a picture of him up here. You can see him right over here. Those are the hands that did the experiment that I just showed you. So we did one other set of confirmations. So we looked at the interleukins to confirm that they worked the same way as NF-kappa B, and in fact they do. And again, just to emphasize this point, these cytokines are the chemical information that programs the T cell and tells it exactly where to go and what to do. So in each case, we see that in our heterodimer, again, this underscore linkage, we see much higher activity, statistically significant higher activity than either the mixture or either of the molecules on their own. So not only are we activating NF-kappa B, but we are in fact eliciting an immune response and directing the immune system to program a Th1 type response, which gives you more cytotoxic T cells. So with my last couple of minutes here, I want to highlight one other area that we've been working on, which is this area of caged agonists, where we've taken chemical tools that allow us to manipulate the way that these molecules interact with the receptors, allowing us to turn the receptor on and off remotely with light. What motivated us is that for each disease, you have to have a new code, a new and different code necessarily. And so how do you find these codes other than the sort of laborious way we'd been doing it, which was to synthesize them one at a time? And given that, if we had all the combinations, it would take us thousands of different molecules and a lot of manpower. And as you can see, Rock's only one person, so that wasn't going to work. So the other way we thought we could find them is if we could selectively activate the different receptors at different points in space and time, we could play the code, we could communicate the code effectively just by turning on the receptors. So this was the challenge. We still needed to find these codes for effective adjuvanticity of things like Ebola. And so we needed a way to do this effectively. So the goal, right, if you're going to develop a wanted poster for any of these diseases is you have to ask the basic questions of any crime, which are the who, what, where, when, and why. And so what having caged agonists would allow us to do in reconstructing these codes is answer the where and the when. We already know the who, which is the Ebola, malaria, HIV, but what we need to know is the where and when. How do we activate the immune system, and when do we do it? And so we came up with this technique, which we call trigger. chemists, we get tired of saying these very long sentences over and over again, so we come up with acronyms for different approaches. And so this stands for Tagged and Remotely Induced Guided Immune Response. But it's actually a backronym, right? We invented it because what we were doing effectively is we were adding a molecule and then reaching into the cellular milieu and triggering that specific cell or that specific molecule and activating it. So the idea is that we take an unmodified dendritic cell with this receptor, we create a molecule that binds to that receptor effectively but doesn't activate the receptor. That's the key part. It stays quiescent, it stays inactive until we hit it with light, at which point we turn on the molecule and turn on the receptor and activate it. So we call this process trigger, where we can remotely trigger an immune response. And we have a whole palette of molecules we can do this with because, again, we sit at this interface between the dendritic cells, which are our gateway to the antibody response, and these molecular activators, which are the key elements of communicating the right code. So could we use light, this tool, to type out an effective code? Can we turn on receptors effectively in different parts of the cell? Can we turn them on at the cell surface? Can we then see how activation selectively at different parts of the cell leads to the type of responses that are necessary for an effective vaccine response? So we can, as I said, if we have two wavelengths of light and the ability to activate these independently, we can create pulsed sequences that are both spatial and temporal. And so it really begins to look an awful lot like a set of musical notes. So if you just press the right keys in the right order, you can begin to get the type of responses you want. So we can have a conversation with the immune system. So this is the chemistry. This is how we actually do it. What we use are these photo cages. This molecule here is called an orthonitrobenzyl group. And what orthonitrobenzyl groups do is that they are great at leaving when you shine light on them. So if you create this molecule, which we did, we figured out exactly where to protect it, and we can uncage it and reveal the parent molecule, which causes dimerization of the receptor, ultimately leading to immune activation. And in fact, we didn't just do this blindly, right? We actually went in and figured out exactly where to put this. So these are the toll-like receptors. This is toll-like receptor 7 and 8. And what we're zooming in on here is the binding pocket of toll-like receptor 7 and 8 and how it's associating with this molecule, this imidazoquinoline. And so I froze it here for you so you can see these are the two receptors. The hydrogen bonding network that is involved in the association and activation of the inflammatory response really all comes down to this C4 nitrogen. This one nitrogen on this one molecule is the difference between no immune response and a massive inflammatory response. And so what we are able to do is by caging or masking this nitrogen, we can modulate the signal and control it so it turned on only when we wanted it to, and only in the place that we wanted it to. So that was the idea. By caging this nitrogen, we can control effectively when and where this process happens and when and where that receptor turns on. So that's the idea. We can activate TLR7 and 8, and here's the data that proves that works. Again, we use that same NF-kappa B assay, just measuring the amount of blueness in the solution, and what you're looking at here are 3 experiments. So we have a control where we add nothing. Here's the parent molecule added without any cage. Here's the caged molecule without any light. And then in orange here, we return that signal when we add light when we shine light on our solution, we remove this cage and return it back to its parent compound, and so we can effectively modulate the signal using light. And that experiment was done by Nari, who is right in front here. And the last experiment that I'll show you here is just one that mostly is just showing off, but we thought was pretty cool. So dendritic cells, what's really fascinating about them is that when they become active, they immediately start trying to engulf everything else around them. And this makes sense because the code is telling them, hey, there's something here you need to get rid of, but they don't know what that something is yet, right? They still need their antigen, they still need that polypeptide to track it down. And so they just begin swallowing everything around them as fast as they can. And this process is called macropinocytosis. So what you're looking at, this dark shadow here, is the dendritic cell, and these green blobs are the macropinosomes that dendritic cell is swallowing of the surrounding solution. And so what we figured out is that we could control when and how macropinocytosis turns on by using light. And so I'll just show you the 3 different examples here. So in this upper left corner, we have the cage all on its own, no light added. And you can see that the dendritic cells mostly just sit there. They remain pretty much exactly the same. You see a few of these dendrites forming, but really no macropinocytosis. Now contrast that with imiquimod, our agonist that's activating TLR7 and 8. You can see these cells are highly motile. They're trying to endocytose everything around them. And this guy, as you'll note, tries to eat his neighbor. And this, in fact, is very common for dendritic cells. It's called frustrated macropinocytosis. And the last thing is when we take our caged compound and we shine light on it, you can see that we recapitulate the ability for these cells to undergo macropinocytosis. So here you can see this cell is macropinocytosing quite a bit, as well as some of the ones in the upper right-hand corner. Not quite as mobile as just the compound on its own, but we can recapitulate the same function. So this is just those pieces of data frozen now, so you can see the difference between the compound, the caged compound, and the caged compound liberated. And so the liberated caged compound looks much more like the active form than does the completely inactive. So we can turn on macropinocytosis and turn on toll-like receptor signaling using light as our tool. And this experiment was done by Lisa Stutz, who's also at this front table. And I should just highlight that of all the experiments I've shown you, this one took the longest. You can tell that these videos this just doesn't happen on its own, right? So Lisa spent hours and hours at the microscope staring into darkness, looking for a beautiful image to show you today. As I'm running out of time, I'll skip over this. This is basically the same thing, again, confirming that we can activate these with light. Now we can show that we can spatially control the cell expression of different interleukins, controlling where we turn on the signal. So by masking and activating only in one position, we can control where exactly we turn on the inflammatory response. So to summarize, hopefully I've sort of motivated why we're interested in this problem, the challenge of creating codes that allow you to talk to the immune system and speak the molecular language necessary to adjuvant and direct a particular vaccine, why we need those codes so desperately, and then our two approaches to the problem, how we're creating those codes synthetically using organic chemistry, and then using organic chemistry to control where and when we turn on a given molecule, allowing us to create a sort of symphony of codes, selectively activating them in the sequence and manner we want to. So with that, I'd like to acknowledge, I acknowledged people along the way, but there's two other people who've contributed tremendously to this project that I haven't had a chance to speak to today. So Janine Tom, who's also in the audience as well. Janine, could you highlight yourself? Janine was the first graduate student to work with me on this project. And so she came into a lab with literally nothing in it and just this core idea, and developed a lot of the tools and techniques that I showed you along the way here. And so a lot of the credit for all of this goes to her. And then recently we've been lucky enough, due to the support of the NIH, to add an assistant here, Troy Moore, who's just helped us sort of catalyze all of the different things that we're doing in lab and make our experiments go faster. And so all of this work, as Ken alluded to earlier, was supported by this NIH New Innovator Award last year. And the goal of this award is to give people money to try things that other people wouldn't try. Essentially, it's the Department of Untried New Ideas, right? And so we were very fortunate to receive that award and use it. We were putting the money to really good work, trying to push forward these concepts of activating and stimulating different codes. And the rest of these agencies also support different aspects of my lab. As Ken alluded to, the DARPA Young Faculty Award supports a different area, as well as the AFOSR Young Investigator Award, which supports work in our microvasculature. So I have all of them to thank, as well as especially all of the graduate students and postdocs I've been fortunate enough to work with. And I'd like to thank all of you for the opportunity to come and speak here. I'd be happy to answer any questions.
Oh my goodness. Any questions for Aaron?
Yeah, so the question was basically, to formalize it, I think you're asking how fast can you get in between each of these different activation moments, how fast between turning on 2 different receptors, and what does the speed you need and what is the speed that gets you anything interesting, right? And so that's something that no one knows the answer to. So it's a good question, and one that we're very interested in because there hasn't been a tool that's allowed you to modulate that response at the time scale that we now have. And so no one knows the answer to that question. I don't know the answer, but it's something that we're actively pursuing. The best I can tell you is that the fastest anyone has ever clocked a dendritic cell at turning on is about 10 minutes. And so the process is somewhere between 10 minutes and a microsecond, but that's about as far as we know at this point. So we're really interested in knowing, is it somewhere in the microsecond range? Is it in the second range? Is it in the minute range? Because that seems really critical for eliciting the type of response you want. So that's what we're hoping these probes are gonna, maybe in 2 years I can come back and tell you the answer, hopefully. It's a good question also. So the question was basically, how will this work? Will it go into living creatures, people, or will it only work in a petri dish? So will this particular cage work in a person? The answer is probably no. Will these types of approaches where we can modulate the response effectively work in a person? Absolutely. And so in fact, we're already in the early stages of working on this in mice, where we're trying to take these compounds and put them into mice and then selectively turn on different lymph nodes. The nice thing about your immune system and in terms of light penetration is that it's mostly on your skin. So your skin is your biggest immune organ. And so in fact, there are already lasers that are approved by the FDA to be used mostly for facial reconstructive surgery and plastic surgery, but could easily be repurposed for activating and turning on these responses on the skin, which is where a lot of vaccines already work. So you could think about pulsing responses that over time, modulating the immune system in ways that it's never been modulated before, sort of what we call micro boosting, and so all of these things are possible with this technology. We just need the person power to be able to execute a lot of it. So I think that's the challenge right now, is not the technical challenges so much as the number of people and the number of hours until we can make something like that work, if that makes sense. Yeah, so the question was, why do you need this level of control versus just, say, adding the molecule on its own and letting it swim wherever it wants? And is that because it's toxic? And so I would say, yeah, big picture, the problem with a lot of these is some of these compounds have already gone into, so we're obviously not the only people in the world interested in this problem, right? And so some of these compounds have already gone into clinical trials as adjuvants for things in cancer or as adjuvants in HIV. And every time, the reason they fail is because they diffuse too quickly and they move around the rest of the body. So you get this really quick response, you get a strong fever, and then nothing happens afterwards. So being able to turn them on selectively and control them and where they go seems to be the key element. And that's what viruses actually do very well, right? Because they reside only in certain places and they stay persistently and presently there, they actually are communicating very effectively with a small number of dendritic cells rather than your whole body for a short amount of time. So yes, controlling the where and the when is turning out to be the key element of eliciting the right type of immune response rather than just an immune response. Oh, that's a very good question. I'm not so the question was, how do you find these people who are naturally immune to things? And I'm probably not the best suited to answer this question, but my colleague in immunology, whose name is Phil Felgner, has developed this technology, just to sort of preface that. But what he does is essentially you go to places where lots and lots of people are getting the disease, you just draw everybody's blood, and then you realize that, oh, this one person, everybody has antibodies, and this one person has completely different antibodies than everybody else. And they've been living with, say, 20 people with Ebola for, 5 weeks and they haven't gotten Ebola. And then you can figure out, oh, when you stimulate them, they produce an entirely different set of antibodies. And so you basically pull their blood and then you analyze the blood to find the difference between them and everybody else. And Phil's developed a technology specifically for doing that, a microarray that lets you read out the specific antibodies in a blood sample. So the question was, has any of this been used to treat Ebola yet?
And these experimental.
And yeah, the answer is probably no. There may be a few people privately experimenting with it, but no one who is doing it in a clinical trial setting. And the reason, the reason is because before these molecules are approved broadly as adjuvants by the FDA, you're not legally allowed to use them on a human being. Oh, so the question is, what is this? So they referred to the secret serum.
Yeah.
So the question was, there were a few people who got Ebola, and the news sort of referred to it as a secret serum. And what it was, in fact, is it was a set of 3 different antibodies that they had grown up painstakingly, and essentially they had about 100 total inoculations of this stuff. So they grew those antibodies. So they're, again, the same thing. They already know which antigens to use. The problem is getting the person's immune system to attack those specific antigens. So if you add those antibodies, that cocktail, that secret serum, you can cripple Ebola or allow the immune system to clear it. The problem is that those antibodies are too expensive to produce en masse to treat, say, millions of people. You could only treat 2 or 100. I think they're basically out now. So, yeah.
Okay, with that, I think we'll thank Professor Esser-Kahn for spectacular lecture. Thank you.
Thank you.